Hello everyone, my name is Ellie Scopes and I'm here today to talk to you about calculating small mammal density estimates using my master's thesis on Guernsey as a case study, and this took place when I was studying for my master's at Imperial College London. Density estimates provide vital baselines for conservation, yet the latest population review by the Mammal Society identified a lack of data for several small mammal species. Furthermore, traditional mark-recapture calculations can be inaccurate, as the surveyor often has to guess the area that is being surveyed in order to get a density. Therefore, new models called spatially explicit capture-recapture models have been designed to take into account space and help to improve the accuracy of density estimates. In this presentation, I will present a modern method that takes into account the position of traps to calculate a more accurate estimate of density, and I will illustrate this technique using my master's thesis, where I calculated a population estimate for the Guernsey vole. The Guernsey vole is a unique island subspecies of the common vole, which has often been overlooked in research and has only really been studied during the 1960s. It is endemic to Guernsey and is related as to the Orkney subspecies of common vole as well. My study took place in the summer of 2018, where I was on Guernsey for 3 months. I used normal trapping procedures, so this means using Longworth live mammal traps placed in grids with bait and hay to attract small mammals, and then these were checked in the morning and the evening. However, there were a few changes that I had to make in order to get the data necessary for this new methodology. First, each individual animal needs a unique mark so it can be tracked. This can be done with marks in multiple areas, such as shown here with these 4 locations, 4 fur clipping marks, where they can then be used in different combinations to get 16 unique individuals. Second, this model is spatially explicit and so requires that each trap has an ID and a coordinate. This can be GPS or grid reference, or you can just use your own system based on the distance between traps, such as shown here. So each of these traps is 5 meters apart, and you can see they've been given coordinates based on where they are in the grid. And finally, you need a little bit more time in your survey than is traditionally needed for mark recapture studies. And this is all in order to get enough recapture to fit the model. So I did my trapping on Guernsey for 5 days, checking in the morning and evening, so 9 sessions overall. And sometimes I didn't get enough data even from this length of survey. So the model estimates the location of home range centres based on the frequency that individuals are seen. An individual that is seen frequency is likely to have a home range near the trap. As you can see from this graph, the capture probability is increased as the distance between the home range centre and the trap is small. The model runs thousands of simulations with different centre positions until it finds the position that best fits the data. And then from this, the density and home range size can be estimated from the centre locations. For my thesis, we had several sites and habitats, so I ran a model for each site and then averaged over the same habitat type. And this gave us density estimates for the different habitats. So the output of the model produces densities and home range for each survey site and for each species. For example, these are the densities of the Guernsey vole in improved grassland habitat from a spatially explicit capture-recapture model. These densities can then be used to produce a population estimate, commonly by using the area of a habitat that you have a density for and then scaling up to get a population estimate. So for Guernsey, I was able to do this because they have a habitat survey for the entire island. And so I was able to generate a population estimate for the Guernsey vole of 130,802 voles on the island. So in conclusion, accurate density estimates are vital for conservation, and new methods of calculating them, such as spatially explicit capture-recapture models that I have talked through today, can help fill this need. As my master's thesis demonstrates, this model can be applied to small mammals relatively easily, to produce density and population estimates for understudied small mammal species. I hope that these models can be used more in the future, and perhaps the Cornwall Mammal Group will consider using them during their own surveys as well. Thank you very much for listening today.